Good morning. My name is Eriocaulon. I'm a postdoc of season pharmaceutical product in China. And I've been a member of this conference since I was in New Zealand in 2014. And today I'm here to talk about our preliminary results of the project to study molecular phylogeny of Okay, now let me explain some about what I'm doing. I've been working on and then interested in the aquatic recurrent plants since I was a student in Japan. And the aquatic plants, which are not quite good, but you can see some lineages in phylogeny of the sperm. For instance, water lily in the beta angiosperm and some pondweed and seaweed and some aquatic plant in the concourse and some regularium and oils. And then today's focus is on the genus Eriocaulon, which belong to Flores here. And most of the species of Eriocaulon has terrestrial form, wetland species, but sometimes you can see here this one. Species have aquatic form or aquatic species, let's say submerged leaves, which explain later. Okay, so Eriocaulon is aquatic wetland. So let's see how the species look like. And here you can see diversity of Eriocaulon morphology. And I will explain some about differences. For instance, here, as I mentioned, most of the species have this kind of terrestrial form, but sometimes the species have submerged leaves or aquatic habitat. And you can see a difference in the number of flower head in the scale. For instance, this species, Eriocaulon truncata, has 1 or 2 flower head, but in this case, Eriocaulon st could be a new species from Laos or something. Is multiple flower head by individual. This is another difference. And you can also see the difference in leaf morphology. You can see here, this one, Rakiana has hairy leaves, but this one, Bonense, has globulous, non-hairy leaves. And in most cases, however, people use the floral or inflorescence characters. And this one, for instance, Eriocaulon sexangulare has the floral leathery flower head. But this one, in the case of this one, Eriocaulon T43, this one has the softy and then much hairy inflorescence. And then in the field, you can sometimes encounter this kind of attractive remarkable species like Eriocaulon echinuratum or Eriocaulon gerianum. The thing is, in the case of this one, for instance, echinuratum, what you can see here, like the spiny one, is because all the flowers has a sepal, which has elongated and narrowly elongated one, so that the flower head is itself is like this. But in the case of Nuclearianum, what you can see here is the enlarged involucral tract, the outer layer of inflorescence, and the flower head itself is here. So that's the difference. And then there are many important morphological characters in flowers. For instance, here, female flower has sometimes 3 stigmas or sometimes 2. And then the other one is the presence or absence of sepal. This case shows only petals, but here you can petals. And then the other one is a male flower in which sepal are segregated like this or tubular. based on these morphological characters, we have around 400 species worldwide. So next slide, I'll show you what's the interesting why Eriocaulon is studied. And the first question is biogeography. And what you can see here is the specimen record of harboring specimen record of family Eriocaulaceae on top and then genus Eriocaulon on the bottom. The difference is like the density of the dot on the Latin America, South America, which is because this continent accommodates many 8 genera of family Eriocaulaceae. But for instance, Asia, Australia, there's only Eriocaulon. So let's see the species number, species difference for the Eriocaulon. And unfortunately, we don't have any reliable literature from the Latin America or tropical Asia. But at least we can say, as for instance, flora Japan has 14 species, flora China 35, and flora India 35, Australia 40, and then some many species are from Africa. So the question is, the Eriocaulon area of origin in Africa, Asia, or Australia. And we have prior information about the biogeography because in 2010, Brazilian group published this paper on which phylogenetic analysis of Eriocaulaceae, family Eriocaulaceae, and which include 5 specimen species of Eriocaulon. Now let's see where the species are. Here it is. Eriocaulon is monophyletic and placed here, and you can see our group, the sister group, this Eriocaulon, genus Eriocaulon, which is genus Mesanthemum, endemic to tropical Africa. So we have an hypothesis. Eriocaulon is African origin, which we will test in this study. And next question is habitat variation. And as you can see here, most of the species of Eriocaulaceae have such kind of wetland form, and the leaves are robust. And yeah, you can see here. And in the case of genus Eriocaulon, you can see not only these kind of species, but also aquatic species, like this one, Eriocaulon nampoense or Eriocaulon scaccidi, which have actually aquatic habitat, and the leaves are not robust, but instead ribbon-like leaves, which could be adapted to this aquatic habitat. So the question here is, how many transition to aquatic habitat? And the last question is polyploidy. As Professor Mark Chase and Sarmashu already addressed, that polyploidy played an important role in the evolution of understanding. And it seems that Eriocaulon is one of the case studies. What you can see here is the data from literature. And you can see chromosome number variation from 18 to 110. Huge variation, and I just show the expected base number of this. So here the question is, Eriocaulon polyploidy evolved DT3, and if so, what kind of mechanism in evolutionary trial? Okay, objectives and approach to the study. We have 3 objectives. The first one is biogeographic origin of Eriocaulon. The second one, habitat transition to aquatic. And the last one is polyploidy evolution. And the approach of this study is not really expensive. It's also one phylogenetic framework using molecular phylogeny. Okay, so method. I should briefly explain taxon sampling. Our taxon sampling covers not all the distribution, but covers most of the important areas, such as Africa. We have 14 specimens sampled, probably representing around 25 species. And the other one includes the Thailand collection, 25, or Cambodia and Thailand here, Cambodia, 20 specimen, and Hong Kong, China, we have some collection, and then the Japanese specimen, and then some are from Ecuador, because we have a person from Denmark, Office University. And we do have collaborator South Africa and Australia. Then these specimens to be added in the future, not today. So in total, our toxin sampling covers 138 specimens, are samples representing around 60 species, not 400 unfortunately. Okay, and the method of choice. What we did is quite orthodox and not very special. DNA markers, we choose first the DNA, multiple RBC, RPOB, gene sequences. Probably we will add one more RPOC1 in the future. And then we try to use the low copy nuclear DNA, in this case 5 from C. It's just start from under progress. And the phylogenetic reconstruction, we did likelihood method and Bayesian method in the software. And we also conducted Bayesian species tree method using the software studies. And then, here it is. The ancestral area character inferences. We did the Bayesian method, Parsimony method, and Bayesian method. Okay, so let's see what we found. This one shows the Mr. tree. Is cluster DNA, multiple RBC, and then you can see Eriocaulaceae species, samples 138, and then we got some well-supported clay like this, which features shown in red, like this clay, and here Asian clay, and species. But some clay are not really significantly supported like this, which are shown in color blue. And here is the 5.C3, and as I mentioned, we haven't got any sequence data so far. So today I don't want to use these, and then use only the cross-DNA sequence data for the sequence analysis. And then what you can see here is the Bayesian star-based species tree. I selected 60 species. And then all the duplicated specimens are into one TU. And you can see the outgroup. This one is Eriocaulaceae, another family, and then here the Eriocaulaceae genera is here. And then later we manually collapse the tree because some some clay have failed to get higher support, so that we use this polytomic topology with higher support for the subsequent analysis, like phylogenetic analysis. Okay, so let's move on to discussion. I said we have 3 questions, and the first one is phylogenetic. And you can see the origin of the species in color, like the blue means the African origin, and then purple means Asian, and then orange means American species. And the thing is that even though the outgroup, sister genus, is from tropical Africa, in the genus Eriocaulon, the early divergent species actually from Asia, like Eriocaulon sexangulare, Eriocaulon spirata. These species are in most cases big in size. So we can say this Eriocaulon is actually Asian origin and not African origin. And the second question is the habitat shift. And you can see 3 aquatic species here and here. So, and the species like this. This is not really aquatic, but other pictures show that sometimes some more photos attached. So Eriocaulon has a multiple transition to occur, probably 3 times, but could be twice, but anyway, multiple transition. And the last question is polyploidy. And then I got data from literature. Say this species have 36 chromosomes, 32 chromosomes species here, and 32, 64, 24, 24, 80, and it's kind of chaos at this moment. It's probably because what we have is the literature data, and so that we start doing the chromosome observation using our own collection. So in the future we can get more clear, I don't know which kind of clear evolutionary history. But at this moment, what I can say is polyploid history is still to be studied in detail. Okay, the summary of this article's talk. We have 3 questions, and then for the biogeography of Eriocaulon, we can say Asian origin is for the genus, and then probably dispersal from the Africa. For the second question, habitat shifting in Eriocaulon, multiple transition to aquatic from terrestrial wetland species. And the last question, unfortunately, we can say anything at this moment to be studied in detail. And the last one is a small topic, future prospect. This one go back to the original phylogeny tree, and what we found is the little variation among the species in Japan.